Hello, everyone, and welcome to today's live presentation, Polypharmacy, a healthcare dilemma with a genomic medicine solution, presented by Roland Valdes, Jr., Ph.D., Professor-in-Chief, Clinical Chemistry and Toxicology Department of Pathology and Laboratory Medicine, University of Louisville School of Medicine. I am Christina Mahalik of LabRoots, and I'll be your moderator for today's event. We are delighted to bring you this educational web seminar presented by LabRoots. LabRoots is the leading scientific social networking website and producer of educational virtual events and webinars. Before we begin, I'd like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. Just click on the Ask a Question box located on the far left of your screen and type your questions into the drop-down box that appears on the screen. We'll answer as many questions as we have time for at the end of the presentation. If you have trouble seeing or hearing the presentation, please click on the Help Desk button located in the promotional board at the bottom center of your screen, or use the Ask a Question box to let us know you're having a problem. This presentation is educational and thus offers continuing education credits. Please click on the Continuing Education Credits tab located in the top right corner of the presentation window and follow the process to obtain your credits. Please join me in welcoming Dr. Valdes. I will now turn the presentation over to him. Dr. Valdes, the floor is yours.
Thank you very much, Christina, for the introduction, and good day to all of you in the audience. I'm very pleased to be here today and fortunate to have the opportunity to pay attention to some of the subjects that I'm going to be discussing. And I'm always very excited about talking about how the clinical laboratory can play a role, can assist in the concept of polypharmacy and pharmacotherapy. And I bet that many of you, if not yourselves or your relatives or loved ones, are in a situation where they're taking many medicines simultaneously on a daily basis. And that's considered to be polypharmacy if it's more than 3 or 4 medicines a day. And, it's an issue that I think is a growing concern. And I believe that the clinical laboratory can have a strong impact on how this might be managed going forward. So first of all, let me just point out that I have no specific conflicts of interest related to this particular presentation. I'm employed by the University of Louisville, funded by the NIH for much of my work, etc. Okay, so what will we be discussing today in the next 40 minutes or so? There are 4 topics I really would like to go over. One of them is precision versus personalized medicine. Those are 2 words that I'm going to try to describe for you because I know that sometimes they can be confusing. The other issue is, what is polypharmacy and how much of a problem is it really for us to tackle? The third one has to do with pharmacogenomics as a potential solution for this problem, and then some aspects about how to implement programs that might actually help to relieve the problem using clinical laboratory science. So let's get started first by looking at what we consider to give, if you look at the future of medicine, one of the most disruptive transformations that are taking place really has less to do with who's going to pay for medical care and how they're distributed. I believe that it fundamentally has to do with the transition from intuitive medicine to a precision medicine practice. And this is written about very nicely in a textbook that I'm going to show you in just a second, but the 2 words that typically come up in the discussion and in the literature are precision medicine and personalized medicine. So the real question is, what do they mean? And we'll describe those in a little bit more detail. Precision medicine really deals with the accuracy and precision of a diagnosis. That is, how much do we know about the disease-causing process? What is the mechanism of the disease? That links precision medicine to its definition. Personalized medicine, on the other hand, really deals with a therapeutic approach tailored to a specific individual, not to a group of individuals. Now, let me take you to the next slide so I can show you and give you a perspective of what I think this really means. This is taken from a textbook that was put together by Dr. Christensen at Harvard, and he does explain a lot of other things that I would recommend you try to assess. It's a textbook published in 2009, very relevant now. And what this shows is a figure of a scattergram, a current map of common medical conditions. And when you look at these conditions, they're on two axes. You've got the x-axis, which is the horizontal axis. And on that axis, you have basically the extent of understanding the mechanism causing a disease. And it goes from the left-hand side intuitive to the right-hand side precise. The more you are to the right, the more precise you know what causes the disease. On the other hand, the other axis, the y-axis rather, the vertical axis, again, goes from intuitive to precise, going up, and it deals with the extent of treatment efficacy. That is basically how well does a treatment work. And these different medical conditions can be placed in various locations and quadrants along these axes. And if you look carefully here, you're going to find that there are four areas that are important. For example, take appendicitis on the left-hand side. There's very little known about what causes the condition, but it's very treatable. You can treat it. You remove the appendix, and the treatment is very efficacious. Depression, on the other hand, on the bottom there, is both intuitive. From a mechanism perspective, we don't know what causes it, and we certainly are very ineffective at treating it. And then on the right-hand side, on the bottom, you have multi-drug resistant tuberculosis, and basically we know what causes it, we know the bugs, but we can't treat it, and that's why it's multi-drug resistance. And then on the top right-hand side is where things get very exciting, and one example is strep throat, okay, where we know basically what causes it, and we know that the treatment is very effective. And this is what leads to rules-based practices and standardization in medicine. So when you look carefully, where you really want to be is way up there on the right-hand side, because if you look at the x-axis, that could represent precision medicine, and on the y-axis, that could represent basically personalized medicine. And they both interplay with each other, and they basically both complement each other and reinforce each other. So that's based on that definition there. I think we need to start looking at the bigger picture. And that is on this slide here, where you look at personalized medicine, what does it mean? Well, you know, this diagram is a circular diagram which basically starts at the bottom with traditional medicine. It defines the pathologic states and the clinical observations. And on either side, on one side, on the right-hand side, you've got personal genomics, which really deals with connecting the genotype to the phenotype and providing insight into the disease, i.e. the precision medicine. Part of this whole thing, the personal genomics. On the left-hand side, you've got therapies that related to drug, pharmacogenomics. And that's where you connect genotype to patient-specific treatments. So when you look at this whole picture, you start getting an understanding as to how precision medicine and personalized medicine might play a role in basically interlocking and reinforcing each other. So let's get on with what we mean by polypharmacy. I want you to look at the right-hand side. This is a figure taken from the New Yorker some years ago. And there's a lady standing in front of her doctor, and she says, I think the dosage needs adjusting. I'm not nearly as happy as the people in the ads. And this is a very common problem. Medicines don't always work, and I'm going to be describing that. So polypharmacy, let's talk about what is the problem, what causes the problem, and what can laboratory medicine possibly provide as solutions. Well, polypharmacy is a national problem. That's the first thing we need to recognize. And it's not recognized as a disease or condition, but perhaps it ought to be because of the great implication. For example, this is literature taken from 2011, 12, and 13. There are over 4.2 billion prescriptions written in the US in 2011. Okay, that's an average of about 13 prescriptions per average American. This is back in 2011. It's probably larger now. 4.1 prescriptions taken by American adults, and over 12 prescriptions are filled by the average American each year. So when you look at the right-hand side, you'll see that 46% of American adults take prescriptions as of at least 2012. The number's probably higher now. 34% of Americans take at least one, and 11% take at least 3 or more prescription medications. And what that means is that 6.5% of American adults are taking 4 or more medicines that are prescribed. If you look at the data that's published in JAMA as early as 2002, this was recognized for years. This is data showing that both men and women, as they progress in age, the number of medications that they're using increases. For example, the dark bar under men, okay, on average says that 50% of men basically total were taking at least one medicine, and approximately 7% on average were taking greater than 5 medicines. But as you increase in age, up through 65, almost 71% or 81% of men and women were taking at least one medicine, and somewhere in the vicinity of 20 to 25 were taking at least 5 prescribed medications. If you look at some additional data coming from our laboratory, this is a study that we did on 30,000 individuals, basically from a retired population. And on the x-axis, you see the number of medications that were being taken per patient. And on the y-axis, basically, the number of patients that were taking that many medicines. And you can see that 92% of the patients were taking 3 or more medications. And this is real data, okay, taken from an organized study that we did. I'm going to show you this a little bit later and what the impact of this really is from the standpoint of polypharmacy and cost effectiveness. So I'll be referring this back in a little while. So what are the cost consequences? Well, polypharmacy is very costly. In 2001, the country spent over $200 billion on prescription medications. In 2008, it was 234 roughly. And in 2012, just 5 years ago, it was over $326 billion of medicines. The other thing is that it has consequences. There's, relative to hospital admissions, if you look at the numbers, 16% of hospital admissions nowadays are related to adverse drug reactions due to medicines. And it's been basically determined that there are over 40 to 70 different potential reactions to an average prescription drug. And you can listen to the TV advertising and they try to fit in, the most 5 or 6 relevant ones. It is the leading cause of death, one of the leading causes of death in the country, and I'll show you some data on that. And frankly, it's been indicated that there are more deaths due to adverse drug reactions than from actually overdosing on administered drugs from illegal drugs. So there's a huge importance and serious consequences. If you look at the adverse drug reactions and the leading cause of death, it's probably considered to be the 4th leading cause of death, with well over 100,000 individuals. And the thing that's interesting is that 60% of medicines in the top 20 list that causing the ADRs, the adverse drug reactions, are linked to genetic variations. And if you look closely at what the FDA box warnings are on many of these medicines, there are over 165 drugs that have FDA box warnings related to genetics. So it gives you a good clue that possibly looking at genetic information might be helpful. Okay, so when you look at the healthcare conundrum, it is a conundrum. Polypharmacy is growing at a very rapid rate. 30 to 40% of elderly patients take at least 5 medications. This leads to drug-drug interactions. We'll be describing those in a little bit. The adverse drug reactions are a leading cause of death in the US. Most of this leads to prescribing cascades, where the first medicine requires multiple subsequent medications, not only for side effects, but because it may not be working. Chronic dependence is also attributed to polypharmacy. And, the numbers suggest that there are well over 2 million hospitalizations annually due to the adverse drug reactions. And I've already indicated that over 100,000 are fatalities. So we understand that polypharmacy is both a healthcare and an economic burden. So then, what causes the problem? Let's focus on that for a couple of minutes. What situations can lead to the use of so many medications? Well, there are several reasons that come to mind, and I'm going to review just a few of them with you. The first one is that drug function is complex. In many situations on the left-hand side, you have an example of what's called a prodrug, where the drug is ingested, and then it requires active metabolism before the actual drug becomes active. That liver takes place typically in the liver. So the drug is not active until it's metabolized. So right away, there's a component where the body has to act on the drug. On the right-hand side, you've got active drugs that are taken, and then those drugs have to be eliminated appropriately through the kidney or the bowels or other mechanisms. So you've got two different situations here that depend on the individual. That's one reason drugs are complex. Secondly, many medicines do not work for their intended purpose. This was an article published in Nature 2 or 3 years ago, 2015, indicating, and the title of the article was an N of 1 in terms of studies, and it's titled Imprecision Medicine. And it basically indicates that for every person that they do help, the ones in blue, okay, the 10 highest grossing drugs in the United States fail to improve the conditions of between 3 and 24 people in red. And the way this data is obtained is through studies where how many individuals does it take to provide a medicine where they take it to then see the given effect that you expect. And you can see that it's very few. Okay, this is for standard medications that are top 10 grossing medicines in the country. Medicines for schizophrenia, for arthritis, for depression, heartburn, high cholesterol, asthma, psoriasis. So this is a potential dilemma. And one of the questions you begin to ask is, why is it that they just don't work that well for their intended purpose? And one operation that you can look at right here is the patient response variability. This takes us into this realm of genetics. So you've got a group of patients in the center that have the same diagnosis. And again, you're assuming that precision medicine is going to be providing a diagnosis that is amenable to the drug that's being provided. If not, it does complicate the situation. But they're all, even though they have the same diagnosis, they're provided the same medicine. So when you start looking at what can happen on the top left-hand side, you've got the medicines are effective and they're not toxic. Or they are toxic in this case. On the bottom left, you've got the medicine is not effective, but it has toxicity. On the bottom right-hand side, you have it's not effective and it's not toxic. So it's totally useless to be able to take them. And then on the right-hand side, you have the group that you want, which is efficacy and no toxicity. And again, let me repeat that there are over 164 drugs listed by the FDA, okay, that are affected by genetics as box drawings on their on the package insert for the medications. So patient response variability could very well be one of the reasons why many medicines do not work. Okay, so the third factor has to do with promoting polypharmacy. This is a lady looking at many, many medicines all at the same time. And this was an article published in the Wall Street Journal back in 2011, indicating what were the hidden influences that sway your medical decisions for using more medicines. And it turns out that advertising was one of them. If you look carefully, that article indicated that for every $1,000 in drug advertising, it translates into 24 new prescriptions by individuals. And that 2/3 of the patients overestimated the benefits of the treatments, and as many as 10 times, many of them believe that it was 10 times more effective than it actually was. So advertising is a very strong component of the push toward more medicines and more drugs in medicine. Another reason that I think is important that adds to this entire complication is that when you look at polypharmacy, it embraces many different multiple conditions. So patients that come to see physicians have more than one clinical condition. A patient that has chronic pain could be due because they have attention deficit disorder or depression. Some who have arrhythmia may have hyperlipidemia and hypertension, also coronary artery disease, and maybe even diabetes combined. So these multiple conditions lead to seeing multiple doctors, which again, many patients have. And then it leads to taking the wrong medicines at the wrong time because you have multiple prescriptions simultaneously. So when you add all of this together, I think I've indicated 4 reasons why polypharmacy is a pretty serious issue relative to whether it can be helped or not. So let's talk a little bit about, can the laboratory, as laboratory medicine discipline, provide some solution? It starts with the idea that I firmly believe is that the role of the clinical laboratory has been always to take bench-side discoveries through to the bedside, and laboratory medicine acts as a catalyst for this process. So the question is, can we provide solutions to this potential problem of polypharmacy? Okay. First thing I want to discuss is the traditional therapeutic drug monitoring approach that most clinical laboratories have done for many years. It's a very valuable approach. And for medications that have a very narrow therapeutic range in terms of concentration in blood, therapeutic drug monitoring, or what is referred to now as therapeutic drug management, or TDM, basically involves giving individuals certain medications and then measuring their concentrations in blood over time to develop exactly how that person is handling that medication. Typically, that measurement is done one time, sometime after the drug is taken, and then sometimes before the drug is administered again, and then adjustments are made. So it gives you information about what the medication is basically doing in blood. And it does give you relationships about the drug in blood. But does it tell you anything about the drug transport, okay, inside the body? Well, not really. And when you start looking at the drug receptors, does TDM monitoring help you with the drug receptors? Actually, no, because it doesn't provide that information that's vitally necessary. So a question you might ask is, can pharmacogenetics link all three of these concepts to come up with a package that might be effective for drug selection and drug dosing? We started my interest in my training program here at the University of Louisville with a review article that we published in Clinical Chemistry in 1997, and it was titled Pharmacogenetics, a laboratory tool for optimizing therapeutic efficiency. And this was published in 97, and then between that and 2010, the National Academy of Clinical Biochemistry asked our group to begin a group to start putting together pharmacogenetics practice guidelines so that they could be addressed from a standpoint of good practice, laboratory medicine practice. And JAMA published a little article in 2006 indicating that they caught wind that we were, in fact, that the Academy was developing some of these laboratory medicine practices. So this kind of started the process. And basically, the whole premise behind this concept was that you've got three legs to a triangle. On the left-hand side, you've got something we call pharmacokinetics, which is how the body alters the drug. On the right-hand side of the triangle, you have pharmacodynamics, which is how the drug affects the body. And the baseline of that triangle that links both of these is something called pharmacogenetics, which is how genetics impacts drug response. Okay, and that was the embodiment of the thinking behind trying to get clinical laboratory information out to help physicians better manage patients. So this had to do with the whole concept. Basically, what it involves is the measurement of SNPs, single nucleotide polymorphisms, okay, to provide individual genotypes specific for a specific medication. And this all transcribes from the idea that, DNA will transcribe RNA, and RNA produces proteins, basically. And those are the proteins that are involved in three different areas of protein of drug handling, okay? And those are metabolism proteins, transport proteins, and receptor proteins. And if you can put genetic information together about those three into a drug response profile, okay, you can get basically something that can help in terms of selecting a medication and selecting the dosing. So I've got listed here a couple of those genes that are involved in the area of metabolism. You've got cytochrome P450 2D6, 2C19. I'll describe a little bit of this a little bit later. Transport proteins are SLO C19, basically, and that particular protein is a protein that transports statins into the liver, and it's associated with myopathy risks. And then you've got the OPRM1 basically gene that's receptor related to the mu-opioid receptor for hydromorphone, morphine, and other opioid antagonists. So the real question is, can you put all of this together as an effective treatment? If you look at cytochrome P450 just for 1 second, the concept here is that most drugs are lipophilic when you take them. And on the right-hand side, you'll see that lipophilic drugs are excreted, or retained, and that retention can lead to a buildup. And what you typically need to do is to biotransform that drug into a hydrophilic metabolite, and that hydrophilic metabolite can then be managed by excretion. And that typically takes place through what were known as cytochrome P450 proteins that exist in the liver and in other parts of the body. And this is important because if you understand what some of these are, you can then characterize individuals, according to this next slide, into different categories in terms of how well they metabolize. And this is only an example of cytochrome P450 phenotypes. You can categorize them typically into either extensive metabolizers, which is really a misnomer. It's really more the normal, most prevalent metabolic situation for patients, or intermediate metabolizers, where maybe they have a copy of one of the genes that makes this particular protein, or a poor metabolizer, where they only have basically 0 copies of the gene and they're simply not producing the protein that's required for metabolism. This particular cytochrome P450 also has a rapid metabolizer status, where you've got multiple copies of the gene, and that leads to very rapid metabolism, which in many cases requires that you give higher doses in order to compete with the metabolism and the excretion. So these little diagrams are very important to try to identify in which category the patient might fit. So from a polypharmacy perspective, you might start to be looking at, well, how many medicines are there that are affected just by 2D6 mediated pathways alone? And there are many, many. I've given you a list here. I don't expect you to remember these, but they fall in the categories of antidepressants, antipsychotic SSRIs, serotonin reuptake inhibitors, beta blockers, opioids, a number of opioids, and many, many other drugs. So this is something that's very ubiquitous relative to the drugs that are metabolized by 2D6, and that's only one of the many SNPs analysis types that can be done in laboratory medicine. So when you begin to look at all of this information, okay, you then begin to think about, well, how do you manage all of this information if you're going to be getting it? And that's where something that I call diagnostic informatics comes into play, where on the left-hand side, you've got molecular information, and on the right-hand side, you've got informatics. And how do you combine all of those to maybe fine-tune personalized medicine? Well, there's several ways of doing this. There's an old way and a new way. Okay? The new model, which deals with the issue of do we explain or do we predict? And on the left-hand side is the older model. This was published by a review in Pharmacogenomics in the year 2000 that basically talks about the explanatory role of pharmacogenetics. This is a role where you make a decision to use a drug. You use a standard drug for that particular condition and a dose. You then have dose adjustments that are based on according to whether the drug is working or whether you have adverse events. And then if you have therapy refractoriness or adverse events, you then do a genotype to see if you can explain what the problem might be for this particular individual. That's an older way. And I think that the modern way of doing this thing is to try to make a decision to use a specific treatment and then do the genotyping. And let that genotyping basically draw and guide the selection of the medicine and the dosing that should be used for that medicine. And then if you have further adjustments, you can then do more sophisticated analysis. Well, the question is, do we test before or do we test after? I believe that the new guidelines are going to indicate as we move along in this discipline that we should test before we take the medicines. And if we do that, then what aspect of this is really the important concept here? That one has to make a connection. You need to make a connection between the genotype and the drug selection and the drug dosage before you can make any intelligent decisions about how to use this, try to predict what medicine to use. Well, how does one do that? I'll give you an example of a drug termed warfarin. It's an anticoagulant that's very prevalently used. It's being used less and less now because there are other ones, but it's one that has a very narrow therapeutic reference range in blood. And because of that, the bleeding, you can get two situations. If on the x-axis, you'll see here the INR measurement, which is the International Normalization Ratio, which needs to be kept as a coagulant indicator somewhere between 2 and 3. That is the optimum way to have that. Basically, a normal human is 1. But if it's much below that, your INR, you have a high odds ratio for stroke. If it's much higher, you can get intracranial bleeding. So it's a very tight therapeutic range for this medicine, and it's very difficult to dose and to select the right timing for the dosing. So this was a good example because there were two issues about this drug that made it a very good model to begin. And it had to do with the two concepts that we've mentioned before, pharmacogenetics on the left and pharmacodynamics on the right. Okay, on the left-hand side, relative to this particular medicine, so you have 40% of the population that are deficient in cytochrome P2C9. Cytochrome P450-2C9 basically involves the metabolism of this particular medication. Now, on the right-hand side, we also understand that over 50% of the population have decreased VCOR, VCOR, which is the receptor for this particular drug, okay, as an enzyme receptor. So if you can combine both of these pieces of information, you might be able to draw actionable information. And the reason you can do this is because if you look at the summary of pharmacogenetic linked functional relationships for warfarin, you'll see that CYP2C9 sets the rate, that is the accumulation or the elimination of the drug. In other words, the clearance. And that's associated with the need for either decreased or increased maintenance dosages. VCOR, on the other hand, the receptor, is involved with the effective concentration of the blood, in blood. And that's the warfarin pharmacodynamic response. It's associated with a specific warfarin maintenance dose that a patient has to have. So how is all of this important? And one of the most ways to look at this that I think is most critical is to look at what happens during metabolism and accumulation of the drug. Okay, this is basically what we call the dynamic approach. And in this figure, I've got three conditions where a patient has three different genotypes for 2C9. The bottom one is 2C9 star 1 star 1, considered to be a prevalent normal. And then you have star 1 star 2, the middle bar, the purple one, and then the green one is star 1 star 3. And what the first thing that you can see, if you look at the S-warfarin concentration in blood on the left-hand axis over time for a specific dosing, you'll see two things. Number one is that as you have a genetic variant of star 1 star 3, you get a large accumulation of the drug. Star 1 star 2 is normal, and you get to the reference range very quickly, basically the red indicators. But what's important here are the little red arrows that indicate when the drug reaches steady state concentration in blood. And you'll notice that for different genotypes, okay, you reach steady state at a different time. So what we've been doing in the past is trying to adjust the INR and the dosing of the drug way before you reach steady state, which is something you don't want to do in clinical pharmacology. Understanding the genetics will understand that it takes a certain amount of time to get to steady state, and it is at that time when you can begin to make adjustments. So if you look at this information, one of the things that was developed, this was one of the work that we did in our university, was trying to develop algorithms that would put this information together. And there are other algorithms available too. And this shows you an example. On the blue, on the y-axis basically is the days after prescribing warfarin with different prescriptions, times. And the blue indicates the actual concentration of warfarin in blood for this one particular patient. And the little squares that are gold indicates basically the INR measurement at the time. And the dosing had to change significantly, and it kept changing. And it took almost 50, 60 days to reach steady state. And this is because the information was not known about the genetic predisposition of this patient, either through 2C9 or VCOR. But when we package the information together and you put it in an algorithm, you're able to get a solution that's basically based on individualized dosing, which means that you can not only pre-select the dosing, but you can pre-select the frequency of the dosing in order to reach a steady state condition within 4 or 5 days, preferably, which is highly advantageous so patients don't have to continue to go back or worry about bleeding or stroke in this situation. So informatics can really put this information together, I think, very valuable. The next question is, well, how do you go about doing this? And if you look carefully at, let me see if I can take you back to this slide here, it shows you this. This shows basically the typical implementation process that you're going to find with just about any laboratory that is doing this kind of testing. Most of the samples that have to be drawn are either blood or the genotyping or buccal swabs. In some cases, you can genotype based on saliva collections. Some laboratories will do that. We use very sophisticated chip array technology now for the arrangement of the SNPs, and we're able to do panels. Most laboratories that are sophisticated at this can actually do panels of SNPs. Okay? And this is important because that panel report then is submitted to the clinician, submitted to the patient, and discussed with the patient, because you'd be surprised how many patients in our experience are very, very delighted to know exactly what's going on relative to their medicines. It helps with compliance and adherence to the medication schedules, and it helps the doctor make a decision about which is the best medicine to use. So what kind of information should be embodied in these reports? It's important that if you're going to be looking, okay, you try to look for information that is actionable, okay, when you have multi-gene interpretations. This is just an example. For example, this is a patient that was going to be provided Celexa as a medication, and this patient, he or she, was a ultra-rapid metabolizer, which increased the metabolic clearance expected with possible increased dose requirements. Okay, but on the other hand, the patient was a poor metabolizer of SLC6A4 phenotype, and that indicates an increased risk of subtherapeutic response and side effects to an SSRI, which is this particular medication. Thus, there are other medications that are suggested. This particular one was not, and you should consider switching to a non-SSRI. This is what the report would indicate, that is not dependent on 2C19, ultra-rapid metabolism, et cetera. So, it is possible to provide valuable information. And a little deeper in this is seen on the next slide, where you're looking at details of multi-gene interpretation that should go along with any of these reports. For example, in the case of a 2C19 rapid metabolizer, you have increased metabolic clearance. I've already mentioned that. Okay, with possible increased dose requirements. And then you have basically responses for, an SLC6A4 poor responder, okay, which means that this medicine is dose increases is not going to be effective. And then you have considerations for non-SSRI selection of medicines also. So in many of these cases, it's important to have the interpretive result very clear up front, and then the rationale as to why, so that doctors can make intelligent decisions. So let's look at a little something. This was a study that was done in our laboratory, and what we did is, in the process of testing patients, we were able to show, that there's a high likelihood of being able to provide actionable guidance. There was guidance in 73% of the patients that were tested. This is a small sample. We took 750 patients that were managed by 27 different doctors with a different range of specialties. And on the bottom axis, you see the number of drug-gene interactions, okay, for moderate, major, or contraindicated severity per patient. And on the left-hand side, the number of patients. So out of 705, only 55 had no indications, minor indications 39, and then the rest had as many as 2, 3, 4, 5, and 7 indications based on genetic difficulties that the patient might run into. And when you look at the data a little bit closer, okay, you can see that of the 27 participating physicians, and the different specialties are listed there: general practitioner, psychiatry, cardiology, pain management, and gerontology. There were 705 patients. Total medications that were being taken were over 8,000, okay, where at least 3,000 had drug-drug interactions, okay, that included the minor interaction for the drugs. And there was total meds with genetic implications were almost 3,000, evenly distributed practically between psychotropics, cardiovascular, pain management, and a multitude of others. When you look carefully at this particular group only, the average number of meds per patient was 12, and the average number of meds per patient with a genetic implication where action could have been taken was 4. So, almost a quarter to a third you can have these implications. Now, I indicated to you that I was going to come back to this particular series of studies that we did here. This is an elderly, it was a retiring population of over 30,000 individuals. And again, on the x-axis are the number of patients, the number of medications that each group of patients were taking. And on the y-axis, the number of patients taken in a particular group. And I've indicated that there were 92% that were taking 3 or more, but take a look at the spectrum here. I mean, you have a very large group taking 7, 9, 11 medicines. You had individuals that were taking upwards of 20, and some were taken in the vicinity of higher than 30. But this has rather important health and financial advantages to this in terms of being able to correct this polypharmacy problem. As an example, in this particular group, if you look at the opiates, analgesics, 46% of patients that were prescribed pro-drug opiates for pain management will not benefit out of the 30,000, 11,000 that were prescribed opiates, 5,000 were subjects that had conflicts. Okay, and this translated into over $100 average cost waste per subject, with an annual expense rate of at least $500, $500,000. And this is only the beginning, because when you started to look at all the other complications in terms of opioids and serotonin reuptake inhibitors and cardiovascular, you notice that 86% of the SSRIs had one or more genetic conflict, and possibly half of those had a single gene conflict, and the other half of those had both the CYP and basically a SL transport receptor conflict too. And then in the case of cardiovascular medications, anywhere from clopidogrel, warfarin, beta blockers, or statin and statins, they all had large percentages of the medications that had gene conflicts. So it's a serious problem that has, potential complications, not only for patients, but the other complication that it has is that it's very costly, and it can be very, very cost savings. We did this study basically that looked at the cost savings that could be substantial because, again, the algorithm we used, we experienced something. We took the same data, and then we extrapolated the advantages of properly using antidepressants, opiates, non-steroidal anti-inflammatories, warfarin, clopidogrel, and polypharmacy related to some others. And the actual cost reduction that were estimated on a per year basis were over 2.2 million savings per 1,000 patients. So when you look at the cost effectiveness of this, you seriously have to consider that some of these pharmacy benefit programs, have thousands of patients. And if they could save this much money every year, using pharmacogenetic information might be a very good way to start the process of guiding the selection of medicines and the dosing. So when you begin to look at the healthcare effectiveness, it's important to recognize that the first drug selected needs to be the right drug. Medication failures due to either inefficiency or intolerability or both leads to a continuum of risk and to the rise in polypharmacy, which you already know is a condition. So you begin with a wrong drug, you get treatment resistance, you get exacerbations, it goes on to hospitalization, polypharmacy, and then you have costs that are associated with that. So it seems to me like the data strongly suggests that pharmacogenetics as a genomic concept can actually identify patients with high risks of failure and provide actionable guidance for alternative medication management. So along those lines, you want to consider if you are going to be dealing with something like polypharmacy that deals with many conditions, it's important to know whether you've got, appropriate information that's linked to many different conditions. And we have, genetic patterns and panels are available right now for pharmacogenomics, dealing with oncology, cardiovascular panels, psychiatry, very important, respiratory and pain management. And I believe that some infectious diseases and immunology will soon be coming as we discover more SNPs that are specifically related to those diseases and the treatments. So basically, when you stop and think about how is it best to generate this information, well, my thinking at the moment is that it's probably wise if we had a systems approach where you had virtual pharmacotherapy clinics that could be approached vertically with platforms, that use diagnostic informatic models for linking fragmented practices so that doctors could link up online with your specific genetic information to be able to give you specific guidance as to what medicine they should use on you specifically as a person and the dosing, and be able to do that at the time of point of care. That this is a concept that I think really needs to be stepped up a little bit, and it's going to make a huge difference, I believe, and that's my personal opinion about how to roll this out, basically. So let me go back to indicate that if you want more information on how you might be able to reduce polypharmacy using laboratory medicine, there's a recent clinics review articles that were published by myself and my colleague, Dr. Reynolds. And it has a variety of 25 different authors that are contributing chapters. And it's a good review that might give you some information about not only the fundamentals of pharmacogenomics, and I think you can go online and figure out how to get these. These are available online, I believe, through clinics review articles. All right, so let me just take a moment here to review what we've been able to do so far and the things that we've discussed. Firstly, we discussed precision and personalized medicine, what the different definitions for those two are, and how they basically complement each other by linking both diagnostic and therapeutics. Okay. We then looked at polypharmacy and what the problem was in terms of too many medicines are being used, many of them are not functional and actually lead to hospitalization and numerous costs. We also then looked at pharmacogenomics as a potential solution, where clinical laboratories can provide very detailed information that provides guidance for intelligent prescribing and selection of medication. I think there's a future for laboratory medicine to have a very huge impact. And then how to implement the process, the concept of using diagnostic informatics, taking molecular information, combining it with sophisticated informatics algorithms to then provide guidance, specific guidance on a specific patient, taking you back to understanding that you've got to have the precision medicine relative to the diagnosis and then the personalized medicine relative to the actual selection of the medications. So with that, I'd like to thank you for your attendance. The whole idea has to do with transitioning molecular technologies to healthcare. Again, I believe that the clinical labs are a tremendous catalyst for taking molecules and making happy doctors by fine-tuning personalized medicine. Thank you very much for your attention, and possibly we can take some questions if we have some time.
Thank you, Dr. Valdes, for that informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the Ask a Question drop-down box located on the far left of your presentation window. Type your question into the box that appears on the screen and click the Send button. We'll answer as many of your questions as we have time for. So let's get started with our first question. Dr. Valdes, you mentioned several situations that led up to multiple use of meds. Which of those do you feel the clinical laboratory could have the highest impact in terms of helping to reduce multiple drug use?
Oh, yeah, okay. Well, that's a very targeted question, and I think it's a very appropriate one because first thing we need to understand is there are certain things that the laboratory is not going to be able to do. For example, it isn't going to control the amount of advertising that's taking place now in terms of trying to get more medications into patients and into the market. So there's little control there. The other little control that we have are the multiple conditions, basically, that patients might have. And they do have these, and it's very legitimate to prescribe different medicines. So the laboratory certainly cannot control that. But where it can control, okay, is to really impact on, number one, the selection of the medication that may be appropriate for the individual and the right dosing for that medication, okay, in order to avoid medication cascades. So this particular guidance is related to proper selection of medicine and increasing the effectiveness of medicine as we work through this. So I think those are the two main areas that the laboratory can have the most significant impact. I hope that answers your question in terms of the target.
Absolutely. Thank you so much. The next question asks, the issue of paying for the pharmacogenetics tests always comes up. Are these tests covered by insurance at this time?
Well, okay. Well, that is interesting, and this varies, frankly. I mean, I've been in the clinical laboratory business for quite a while, and I know that insurance coverage is always an issue, and sometimes it does fluctuate. I can tell you that right now, CMS through Medicare does cover some of the pharmacogenetic tests. It covers 3 or 4 of the tests, sometimes as high as 5, and it's limited to some of those. Okay. Private payers are a mix. What I've found is that some will pay for some of the tests and others will not. So it's important for the patient to find out whether these are covered or not, by the medication. So I do believe, however, that the cost of the tests are decreasing very rapidly from year to year in terms of being able to provide this diagnostic information. And because of that, I believe that the two things that are happening is that the costs are being reduced in order to provide it. And secondly, there is more and more recognition that, in fact, this is useful information. So my guess is that CMS will begin to cover more of the tests and private payers will follow and cover more. And we also recognize that in the future, CMS is going to be basing their cost basis and their repayments based on the private sector. So I have a feeling that there's going to be unity between the two and that more of these tests will be covered. So yeah, I see a good future for coverage for these tests. And again, the expense is low enough now in some of these situations where patients might be able to cover it themselves through their payment programs.
Excellent. Thank you so much for those insights. It sounds like good news. The next question asks, do you see the value in genotyping all individuals in order to have this information available at times of prescription? And is it feasible cost-wise?
Yeah. Well, yes, it depends, really. I think that, the first thing we have to get over is this fear that genotyping individuals is bad. And this is one of the things that, has had a drawback in the use of this information because many people don't want their genetic information available. But what is important to understand is that the type of information that is obtained by a laboratory for purposes of pharmacogenetic testing, it's not basically indicating anything about a disease process or predicting your future health, et cetera. It's really focused on a very targeted use. And that really does target the information. And because of that, I think that more and more people are realizing that, yeah, I think it is important for us to start getting this information into our medical record. So, the idea that we can genotype individuals early on in life is probably, people have proposed this at birth. It may be a little bit of a stretch at this point, but certainly when you begin to start getting different illnesses and you begin to start aging and requiring more and more attention medically, that might be a good time to actually start thinking about doing genotyping that will put your medicine record on record so that doctors can use it. So, yeah, I think it does make sense to develop platforms that are going to be able to take this information and use it.
Thank you so much for your answer. Appears we have time for one last question today. This question asks, would you recommend testing for panels of pharmacogenomics tests as opposed to selecting specific tests based on anticipated medications?
Yes, I really do. And the reason for that is, as I indicated earlier, one needs to understand that because the combination of genotypes sets a balance for how much medication may be prescribed or which one might be effective, knowing one or two genotypes only is not going to be sufficient. The most sophisticated algorithms use panels. I have found, basically the work that's done by my colleagues in my laboratory, that usually around 12 to 14 genetic tests is what's optimally necessary. It links up with databases about drug-drug interactions, which can also be brought into the algorithms. And those are the main ones, around 12 to 14, that have the best description in the literature in terms of their effects. So I certainly recommend that panels be considered. I think that limiting it to just a few because you're taking one medicine or anticipating taking two really is not optimum in terms of doing the complete balance of how one of these genotypes might counterbalance the effects of the other in terms of the drug selection. I think I alluded to a few examples, and some of those examples in the literature can be seen by some of the references in that little textbook that I recommended. So yes, I do recommend panels over specific genotypes. Yeah, thank you.
Excellent. Well, thank you, Dr. Valdes, for your presentation today. And I would also like to thank LabRoots for making today's educational webcast possible. Before we go, I'd like to let everyone know that today's webcast will be available for on-demand viewing through July of 2018. You will receive an email from LabRoots letting you know when this webcast will be available for replay. Please share that announcement with your colleagues who may have missed today's live event. That's all for now. Thank you for joining us, and we hope to see you here again soon at LabRoots. Thank you and have a great day. Goodbye.